So it's my pleasure to introduce Jamiree Harrison, who is my PhD student from the Biological Control Lab. And he is an incredible scholar, an incredible student, and an incredible colleague. I'm very proud to be able to have the opportunity to advise him. Jamiree started out with a background in mathematics. So he did his undergrad in mathematics at UCSD, and he came to the mechanical engineering PhD program at UCSB and selected as his areas of interest dynamics, controls, and robotics as the focus area, and computational science and engineering as his second focus area. And he's very good at both. Along the way, we managed to convince Jamiree that he might be able to find a lot of interesting problems to look at in theory and in computation in synthetic biology. And so it was Jamiree was an exceptional student from the start. Within, I think, the first year or the two, he already published a paper, a journal paper in mathematics, outlining essentially methods for identifiability of Koopman operators with toggle switch models and genetic circuits. And so he might talk about that today. But more recently, after finishing that initial work, he turned to a very hard seminal problem that I would say has been skirted around by synthetic biologists for a long time, which is the engineering of genetic circuits that have time varying behavior. And so I'll let Jamiree talk through the details of why this matters. But in essence, most biological parts that we develop in synthetic biology are only statically characterized. They're statically characterized in the sense that fixed parameters are used to associate with every single part that we have. So when you think of this, like when you look at like a resistor or a capacitor, there are fixed static constants associated with their performance. In biology, these things are actually all fluctuating moieties. And Jamiree has introduced a new branch of computation and a series of experiments to interrogate the dynamics of these circuits. So with that, without further ado, I introduce to you Jamiree Harrison.
Cool. Thanks so much for the introduction, Enoch. And first, I'd like to thank my committee for being here. I really appreciate it, all the support over the years through the classes, just through everything. Thanks so much for all your support. One thing about decorum, the committee has free reign to interrupt me at any point, but the general audience, please save any questions for the end. Cool. With that, we'll get started. So the title of my dissertation defense is Parameter Varying Models for Hybrid Motors in Bacterial Genetic Circuits. Today, we're going to start out with some background context for kind of like where this work sits in the context of the field at the intersection of synthetic biology, dynamical systems theory, and control theory. Then we're going to get into some of the results that I came up with in this whole shebang. So without further ado, we're going to get kicked off. So some background context. In 1961, the lac operon was discovered that regulates genes in E. coli for metabolizing lactose. This type of gene regulation in bacteria was a seminal piece of work that leads to all kinds of different types of gene regulation studies that have come out since then. After this, we have restriction enzymes that were discovered that can chemically cut DNA. And that process is used in PCR, which most of you are familiar with in the recent years due to COVID, PCR tests for COVID. You have PCR that you can use to amplify DNA, meaning you can make copies of DNA that's used in cloning for amplifying genetic parts that go into genetic circuits. And then the first synthetic genetic circuits, i.e. the toggle switch and the repressolator, came out in 2000. And these were designed gene regulatory networks that were founded from the principles of math. And that's kind of like essentially where our work sits right at the forefront of, because we basically expand on some of the genetic parts that go into these genetic circuits. And then after this, you get standardized genetic parts, i.e. like biobricks coming out of places like MIT. And these basically allow people to do plug and play kind of maneuvers in setting up genetic circuits. So that way, if I design like a cool part, then someone else can take that part, put it in their gene circuit, and then do cool things with it that you're going to see in a second. And then more recently also, we have CRISPR, which is more gene editing focused kind of things going on. But essentially, I didn't do CRISPR in my projects. I use other assembly methods. So yeah, this is kind of like the background for synthetic biology. And now we're going to get into some of the background for dynamical systems, what they are, basics of controls, and then we're going to get into the heavy stuff. So here is just a basic promoter gene functional group. So a gene is off when it's not being expressed. If you have a promoter upstream of a gene, then the promoter regulates that gene expression. That's essentially it. Here in 2008, I believe, Golden Gate Assembly by Angler was produced, where you can essentially produce genetic circuits in a circularized plasmid all in a one-pot reaction. And this basically allows you to plug and play the genetic circuits that I was talking about previously and stick promoters into destination vectors and then regulate these kind of genetic circuits. So in terms of dynamical systems, if you have a system, just like some system, like a box that you don't really know what it does, and you have states that represent things about that system, this could be anything, right? If you have like a pendulum, right? One of the states could be the position of the pendulum. One of the states could be the velocity of the pendulum swinging, anything like that, right? And then you have system observables, things you can measure, i.e. the position, the velocity, simple things like that. The states may depend on each other. They may be interconnected. You may have some complicated network. Some states may be immeasurable. But basically, any system that changes over time, we just call that a dynamical system. Okay. A system that changes over time will be represented by a differential equation usually, because this term right here represents the rate of change of some states x, right, given by some function. And then y would just be your observables, right, which is some function of your states as well. So in terms of controls, if you take that same dynamical system, right, in our case, we care about bacteria, then what kind of inputs can we design in order to control that biological system? That's kind of the essence of control theory in terms of dynamical systems. Cool. So going back to the types of biobrick genetic parts kind of things, what we're trying to do today is we're trying to expand the toolbox for genetic engineering by providing a library of promoters that regulate gene expression. That's essentially what we're doing here today. So why do we care about this? Well, if we can take DNA, put it in bacteria, then you can get desired output chemicals. So more recently, people have been able to use bacteria to do things like degrade types of plastic called polyethylene. And then also there are bacteria more recently that can produce hydrazine, which is a chemical used in rocket fuel. So if we can design promoters to optimize pathways to produce these chemicals, then we can get more efficient bioproducts. That's essentially the big motivation for this kind of work. Cool. So promoter libraries are a pretty common thing to do in synthetic biology. Like producing a library of parts is a thing to do in SynBio. It's like one of the things that people do. So the Anderson promoter library is one of the foundational promoter libraries that we're going to be using because essentially I take one of the promoters and then edit it in fun ways to get cool phenotypes. Another notable library is the Miano library that came out of the Hasty lab somewhat recently, and they use a promoter library that is inducible to detect different types of heavy metals. So that's kind of like the essence of like biosensing. You can use bacteria and genetically engineer them in order to detect compounds that you may or may not want to have in your system. So with that, if we can take the previous motif of the promoter upstream of a gene that regulates gene expression, what if we take two promoters that activate in different phases of the bacterial life cycle? So ideally stationary phase and growth phase. We're going to get into that in a second. Bacteria have generally four stages of their life cycle. On the left, you're going to see the log number of bacteria, time hours on the x-axis. And you start off with lag phase. This can be thought of in general as if I have bacteria sitting in my freezer and then I inoculate some cell culture, it's going to take some time for that culture to heat up and for those cells to proliferate. They don't just start multiplying immediately, so there's some lag there. After that, then they start exponentially multiplying because cells divide and then they multiply. That's how they go. After exponential growth phase, eventually the rate of their reproduction will meet their death rate, and then their population will stagnate and they will reach kind of the carrying capacity for the environment that they're in. After that, they will use all the nutrients in the lab environment that they're in and they will begin to die. So what we have done is we've designed a library of over 100 hybrid promoters, like the one you saw on the previous slide, that are able to activate gene expression distinctly in both log exponential growth phase and stationary phase, which we have represented by these two different sets of parameters here, because these parameters govern rates of synthesis that you're going to see in our next few slides. What you're seeing right here is relative fluorescence, and then this green curve right here is GFP over OD. This GFP is green fluorescent protein because essentially we can program the cells to produce a protein that glows green. That way we can know exactly what kind of proteins the cells are producing in real time by just tracking how much of the fluorescence is taking place based on that gene. So what you're seeing here basically is an example case, right, of this kind of expression that we're talking about, is that once we hit this switch time, right, from exponential growth phase, stationary phase, at switch time tau, then we are seeing a new rise of expression, right? And this is kind of the characteristic behavior that we were trying to look for, because if we can increase expression throughout the bacterial life cycle, then you can get more bioproducts potentially, right, throughout the entire life cycle of the cell. Cool. Some background into sigma factors, transcription factors, and like how this whole thing works on the plasmid side of the cell. What you're seeing up here in the top left are transcription factors binding to the promoter region. And these are just proteins that float around the cell and they increase, decrease, regulate gene expression, right? Ultimately, they will increase the binding affinity of RNA polymerase, which is an enzyme that reads the DNA sequence and then transcribes it to RNA. The RNA then gets translated into proteins, and that's how you go from DNA that lives inside the cell to proteins that are produced by the cell. Sigma factors are essentially bacteria-specific transcription factors. Transcription factors also exist in mammalian cells, like you have them inside of you and in every cell of your body. But sigma factors in bacteria are dedicated generally to the initial step of transcription in bacteria. And since we have two distinct promoter regions for our hybrid promoter library, these sigma factor dynamics are going to be kind of what drives these different phenotypes throughout the bacterial life cycle. So the role of sigma factors in both of these bacterial life cycle phases has been relatively studied. And one of the main driving forces that we're going to explore, or that drives the work rather, is dual sigma factor preference, which implies that a gene can respond to two different sigma factors depending on the current condition the cell's in, i.e. what phase is the cell currently in? Is it in growth phase or stationary phase? Cool. Also, they don't usually have like one sigma factor. Bacteria have multiple sigma factors depending on what types of things are going on in the cell. They have sigma factors that regulate gene expression in terms of stress response, in terms of nutrient decay, all kinds of things, right? Starvation, different types of things that would help the cell survive, essentially. So an overview of the talk, generally, this is like the big picture, is that we're going to identify both log and stationary phase promoters. We've already identified one so far from the Anderson promoter library. We're going to go more in depth. After that, we're going to mutagenize this hybrid promoter sequence in order to get a library of promoters, right, from 1 through 100. And then we're going to model the behavior of gene expression that is governed by the hybrid promoters. After we do that, we're going to take these models which have the parameters, and then we're going to fit the parameters to the models so that we can reconstruct the phenotypes and understand the math that's going on behind the scenes, right? Understand the dynamics of the circuit. Once we do that, then essentially we would have constructed and parameterized our entire hybrid promoter library. That's essentially the entire goal of this talk and of this work in general. So with the big overview out of the way, all the background out of the way, then let's get into exactly how I did it and what kind of results that we saw. So what you're looking at here is the exact hybrid promoter sequence that we mutated In blue, you're going to see the mutation sites, and notice that they're not the standard GATC that you're used to. They're N, because essentially you can stick any random ATCG in there, and then you get a new hybrid promoter. If you were to do that, then you would get over 1 million possible variants, because you have essentially 10 boxes and then 4 choices to put in each of those boxes. So combinatorially, you would get over 1 million variants. But due to the fact that no one can really check like 1 million variants, like to see what they're going to do, then we checked a subset of 1,600 variants. And then from those 1,600, we got 100 variants that we put in the library. Cool. Yeah, of course.
To help provoke thought
here. Of course.
So you could imagine pooling 1 million possible variants into a single reaction and doing something like bar-coded RNA sequencing to profile expression. So can you talk a little bit about where that breaks down with what you plan to do with this promoter library?
Yeah, sure. So just to reiterate the question, you're saying that I could, in theory, sequence like 1 million of these variants, but you're asking the question of like why I wouldn't do that, essentially?
Yeah, why did you keep everything separated into different wells as opposed to pooling them all into one tube and then barcoding them?
Well, I mean, barcoding 1 million variants would be like relatively infeasible, no?
It's feasible. What I'm driving at, what can help is like you have to think about whether or not you could take time series measurements and dynamics of all of those samples, because each sample would be you'd have to sequence with next generation sequencing the pooled library. So you can. The point is you can barcode and track 10 million, 100 million possible variants. It's feasible, but you lose resolution on a different axis, particularly if you're talking about.
Okay. Thank you. Cool. Another thing to note on this slide is that essentially we ordered the DNA sequence after it being randomized from a company called IDT, essentially, and they sent us a single tube filled with all of the hybrid promoters. And this was a much more like cost-effective, feasible way of doing this reaction, I believe, than the one that Enoch might have mentioned. So once all of these hybrid promoters are sitting in a tube, then we can take them and perform Golden Gate assembly, so that way we can put all of these promoters into backbones. Once we have these promoters into a simple genetic circuit upstream of GFP, so we can track the phenotype expression, then we can put the plasmids in the cells through a process of transformation, and we put them into E. coli. You'll notice here that the plasmids we put into the E. coli are in combination with the genomes that already exist within the cells, because bacteria already have genomes. That's how they exist. But they must take the DNA that we give them, because we put antibiotics in the system, and then we put antibiotic-resistant markers in the backbones. So that way, if they don't take the DNA we give them, they'll die. So it's kind of like extortion, but they don't mind. Cool. After that, suppose that we have all of these bacteria with plasmids in a tube. Then you can take that tube and essentially streak that tube across a plate. And now colonies will grow on that plate, so provided there are sufficient nutrients on that plate. So essentially, there's a petri dish filled with nutrients. It's called an agar plate. And you're seeing that a lot of these colonies are glowing green because they contain the plasmids we put in them that make them produce a green fluorescent protein. So each of these colonies possesses only one plasmid, which contains only one hybrid promoter, because as the colony progresses, the colony grows from like essentially single cells, right? And then it divides, and then you get a colony essentially of a clone of the cell, right? So if you have one cell, it divides and then divides and then divides and then divides. You essentially have 1 million of that single cell. So by picking one colony and then tracking its gene expression in a plate reader experiment, then ultimately you can be assured that you're tracking the phenotype associated with that particular genotype. From here, you pick the colonies, passage them up so that you're not going to get as big of a lag phase in your plate reader experiment. And then you put the plates in a plate reader, which measures the GFP expression over time, which is what you're seeing right here in fluorescence over time. And also, you'll notice from this data set that you can see switching behavior, right, in the GFP over OD signals over here. You have, once it hits stationary phase, you have some going off. You have some getting like weird spikes and that kind of behavior. You have some going low to high. Fun stuff, right? Types of phenotypes that we were looking for. After that, then we can take these phenotypes. We can take the cells that exist within these wells and then send them off to a company that will tell us what sequences exist within these cells, right? That way we can basically validate that the sequence that we thought was in the cell is actually in the cell, right? It's just one more validation step. Cool. So what you're seeing here is OD on, oh, there we go, it was just laggy a little bit, my bad. But what you're seeing right here is OD in red, which is optical density, which is a proxy for the number of cells that are in that given well. And you're seeing, if you just take this guy, for example, you're seeing the cells rise through growth phase, and then they even out because they're in stationary phase, right? They hit their carrying capacity of their environment, as we mentioned before. And then if we take one of these guys, for example, right, you can see that once stationary phase hits, then you get these switching dynamics that we were talking about, right, due to the hybrid promoter gene regulatory network. Cool. What we do is we take GFP over OD on this graph in order to get kind of a GFP per cell kind of metric. And then you can also see the data a little bit more clearly, right, in terms of what we were talking about. This also gives us the ability to look at one data stream when comparing the data to our models, right? Because it's easier to work with one data stream than to generally, if your model is only identifiable with a certain number of parameters, a certain number of data. We'll get into the modeling later, but just FYI. After this, you'll notice that a lot of these phenotypes might look a little bit similar. So what we do is we take the 1600 that we sampled and we cluster them into 100 based on a clustering metric that you can ask about later if you want. But essentially, it's based on the similarity of these phenotypes. How the phenotypes that are similar get clustered together, because ultimately I don't need to track, right, like 3 of these guys, send them off for sequencing, and then pay all that money. I'm just gonna like send off one and then get that one phenotype. So what clustering essentially is if you have data points that are similar, they get clustered together, and then these clusters have centers. If you plot data points, right, you can generally put centers to where those data points are. Here we have a high dimensional space, so you can't actually visualize it, but they still have a center in the time series space that we examine. And then here, we can actually track how similar these phenotypes clusters are to each other. That way, if we have the sequences and the phenotypes of similar clusters, then we can see how similar those genes are as well during the post-analysis, essentially. What you're looking at here are the 100 phenotype clusters that we got due to the data analysis portion. And you'll notice a lot of late stationary phase type dynamics that we were looking for. The black ones that you're seeing are ones that were replicated across multiple plates. And then the ones that you're seeing in purple are some of the more distinct phenotypes that are of interest that we are using essentially for secondary experiments. And then this is the validation via sequencing that we were talking about, because essentially if we ordered the hybrid promoters that were mutagenized from the company and we were expecting randomness, right, at the nucleotide sites that we were expecting, then you'd expect kind of a uniform distribution for all the nucleotide sites that you could possibly expect. And that is what we see generally. So if you look here, you'll see about in all of the designed mutagenized base pair sites, you're seeing about like 25% of each of these nucleotides, roughly, right? And then you're going to get some off-target mutations that occurred, and then those just happen, right, due to nature. Like nature mutates sometimes. So with that, we've hit essentially roughly the halfway point without getting into all the future directions and all that in the talk. So with that, I'll give the committee a chance to ask any questions at this point. If not, then we're going to get into the modeling portion, the algorithm for parameter fitting, and all of that, because so far what we've done is we've showcased the synthetic biology portion essentially for building the hybrid promoter.
So if I understand correctly,
Mhm.
you essentially you're really interested in these different phenotypes, right?
Yeah.
I put it lactose on 1,000,000, you did 1,600, then you clustered, then you got 100.
Mhm.
Did you get every behavior that you were hoping to get?
Good question. We're gonna get into that towards the end because in future directions, yeah. So short answer, some phenotypes are theoretically infeasible, and then trying to elicit as close to infeasibility is kind of like a neat goal to kind of strive towards. Yeah, but short answer, no. There are some interesting phenotypes that we wish we could get. Yeah. Yeah. Cool. So here is a very standard prototypical mRNA protein synthesis differential equation. What you'll see here by this m dot is the rate of change of mRNA. What you're seeing here is the rate of change of the protein, in particular the GFP protein that we were working with. Both of these, of course, are going to be greater than 0 because you can't have negative things. Cool. So what you're seeing here are the rates of synthesis of these respective mRNA and protein. And then also mRNA and proteins, they decay over time. So we have the respective decay rates in this model. And throw back to what Enoch was saying earlier, where we have static models for these kinds of behaviors. We would categorize this as a time invariant system because these coefficients, these alphas and these betas, do not vary with time. But what we see in our behavior is we see phase-dependent behavior due to the protein synthesis, which means that these rates of change are varying with time. So if we try to fit a model with these static rates, you're going to get bad data reconstruction, meaning your model is not actually capturing reality. So if we expand this model to have time varying parameters, then we should be able to better capture the behavior. And spoiler alert, we do, but we're going to talk about it. Cool. So if we suppose that this time varying behavior is phase dependent in the piecewise constant sense, that means that when there undergoes a phase change, right, the parameter changes. And this happens for all of these parameters. Up to some, there's reason to believe under our conditions that we experienced, and then most conditions, that if you're not actually editing the DNA in order to induce decay rate differences, then these generally are going to just be constant the whole way through. And then we'll get into that later. But the most generalized model you could possibly get, right, would be one in which these decay rates also vary with time. Cool. This would give us a linear parameter varying system. And the dynamical systems theory that underpins this is like very rich, well documented. And in understanding this kind of behavior, we can essentially like use all of the literature that that exists in order to examine and analyze these systems. So before we just throw a model at the data, we have to actually analyze the model a little bit to make sure that the model is going to be identifiable. Let's take a brief pause so that I can get rid of an annoying notification on the screen. Thank you. Cool. Okay. So what you're seeing here in the first equation is red. Why is it red? Well, you'll notice from the data that I showed you, we didn't measure mRNA. I showed you protein, I showed you OD, which means that this is one of those immeasurable states that we talked about. Now, generally, this is not immeasurable in like a gram context. There are methods to use to measure RNA. We just didn't do that because essentially it would have made our experiment a little bit harder to work and a little bit less valid, because if you're using a fluorescence marker also to capture mRNA, then those kind of signals could impede with the fluorescence signals that we were trying to measure in the first place. So it's one of those like annoying things about experiments, right? Is that sometimes it's a little bit annoying. But anyway, we have the protein, which we do have measurements for. But then we have this M term that contributes to the rate of synthesis of our protein, right? So what do we do about this? Well, what we can do is we can say we can't measure mRNA, but we probably could infer some of these parameters if we amalgamate them in the following way. So take this alpha M term. If we call this theta, then ultimately we can infer theta under the assumption that we know delta within certain bounds. If we know delta within certain bounds, which we do from the literature because we're using a variation of GFP, which is a superfast folding variant, which is why it's called SFGFP. And we can use these bounds on delta given our data in order to infer the rates of synthesis theta, right? And although we cannot actually truly infer these alpha terms, this alpha M, this beta M, these terms, this is essentially like one of the best that we can do given our experimental setup. Cool. So how do we actually fit these parameters? Well, so we developed an algorithm essentially that does this. There are other input estimation algorithms, but we found that in order to have one that works with phase dependence, and then also one that you can use with assuming some parameters that are constant and then some parameters that are time varying, that it was best that we use an algorithm that kind of uses switch detection involved. So if we have this switching behavior, then if we have an algorithm that can detect these switches and segment the data into intervals and then fit parameters over these intervals, then that would be the best way to go. So if we can segment the data and then have the model that exists over these two time intervals, then we can just numerically integrate our model and then fit the data to the model, right? And then find the optimizing parameters that do this. So this is a basic setup with only one switch, so we get two time intervals. After fitting over this one, and then you find the parameter estimate for your static parameter, then you can use that parameter estimate. You don't need to estimate it again. So in your next step, you can just assume you know delta P1 because you found it in step 3. And that helps with reduce the space of parameters ultimately. And yeah, so we can generalize this method. And in generalizing this method, we can now detect n switches, arbitrarily as many switches in the data that take place. And then using this, we can capture time-varying parameters of more complicated models that are not just ours, but across physics, other fun systems. Here's the fun system, for example, if you have the advection diffusion, PDE, right, with a sinusoidal alpha term and then a constant diffusion coefficient, right, then you can accurately reconstruct the data that you give it, and then also accurately reconstruct the parameter using a sparse regression on the parameter outputs of that we got from the algorithm. Cool. So what we see when we run the algorithm on our data is that if we take this GFP over OD, run the algorithm in order to fit the parameters, what you're seeing here is theta in red and then delta in blue, that we do see time varying theta parameter, right, that governs the rate of synthesis of that protein. And it matches up kind of like with what you would expect, right, in the behavior of this, right? When protein synthesis is decreasing, you get decreased rate of synthesis, right? So on and so forth, right? And then the bounds on the delta allow us to get error bars essentially on our parameter estimates, because we can never be too certain about our parameter estimates. We can only get them within some bounds of uncertainty, right? Cool. So with that, I'm going to walk you through everything we did once again, and then I'm going to get into some future directions about things that we wish to do in the future. And then we're going to do some acknowledgements, close out with some questions. So in terms of the modeling portion, we not only found and built upon the models that already exist in the literature, but we've expanded upon them in a way that was not only necessary, but intuitive given the structure of the genetic circuits that we had, right? After that, we fit parameters in a way that gives us accurate reconstruction of the data that we see. And if you expand upon these models and then take our promoters and then put them in more complicated genetic circuits with more complicated models, you can then fit parameters to those models and then cross-reference. Are the parameters similar or not that we saw in the simplified version? That's kind of like a future direction that we could take, and we're working on it. Cool. Using this, we've built the promoter library, right? So now we have a document where you can scroll through essentially and say, what phenotype do I want? I want the fun looking one. And then you go, what sequence do I need? Okay, I'm going to order that. And then, well, I want to kind of predict what's going to happen when I put it in a simple circuit. It's like, okay, cool. Now you have a method to do that. So this is kind of like a peek at what that document looks like, right? You have the phenotype, the genotype, and then the model isn't written out because essentially you need a picture, right, of that parameter profile that you saw before. But yeah, it wouldn't fit on the screen. So yeah. So that exists for each of these phenotypes, right? Hence the ... because I'm not going to fill the screen with 100 of these guys. Cool. Connecting back to the original motivations, now if you have a circuit that degrades PET, for example, and you want to either increase or decrease the rate of degradation of your plastic under certain conditions, for whatever reason, then you can say, okay, well, what's a simple way I can do that? Instead of designing like a very complicated repressive genetic circuit with like on-off switches that may or may not work and is not robust, you can take one of these promoters, which is just one part, stick it in your already existing genetic circuit, and then potentially see that behavior enacted in your system simply in a predictable way. Same thing with hydrazine, right? That's kind of like the power of promoter libraries in general, and also like genetic parts libraries in general. So for future work, and then bringing it back to Professor Bulow's question, also, we need to do more reproducibility studies in order to make sure that the phenotypes that we're seeing that are very interesting are able to be reproduced basically on command, right? Because it's not very useful if your phenotypes are like 1 in like 100. You need it to get like pretty reliable in order to call it like a reliable part, right? So we could put HPL promoters in more complex inducer-based genetic circuits, such as the toggle switch repressilators, just fun things, right? Just more complicated genetic circuits, and then see if they still exhibit the same type of phenotypes that you would expect under different genes, and then also under different regulatory dynamics caused by other genes. Because right now we just have promoter gene. What does it do, right? What if this gene interacts with this gene? Then what's going to happen, right? What if both of them have time varying dynamics, right? Then what happens? Cool. And then a natural next step also would be to use machine learning not only to map back and forth between these genotypes and phenotypes, but also can you use machine learning in order to basically fill in the gaps? So for example, we sampled 1,600 sequences. What if you could fill in some of the other close to 1 million variants in your model that you trained on our 1,600 variants, and then produce phenotypes that you predict from these sequences, right? Then you can essentially fill out the phenotype space that Professor Bulow was talking about potentially, right? And then you can basically intuit what sequence you would need in order to produce a certain phenotype. That would imply some type of like reliable genotype to phenotype mapping that is happening. And we're working on that currently. A colleague of mine, Alec, has like a pretty decent software that can do that with machine learning for a spacer library for GFP. But we're working on that for the promoter library as well. Another thing would be alternate methods for the parameter estimation. Right now, the parameter estimation seems to be working pretty well, at least up to the old engineering adage of it works good enough, so might as well keep it. But there's always ways to improve in theory, and then coming up with some other type of method could be fruitful under this assumption that we can get more complicated genetic circuits that our algorithm might fail under those conditions. But that's like an unknown type of situation. And then we can explore more of phenotype space. So these are the 1600 phenotypes that we sampled, and they exist in 960 dimensional space because we have 960 measurements over time. So if you could plot that as a vector in 960 dimensional space if you could, right? What we do is we do a 2D projection onto principal components of this data set using PCA. And then we can now plot all of these phenotypes on a 2x2, right? Or on a XY grid, right? And what you're seeing here on this plane are the clusters, right? Are the similar phenotypes in a more interpretable fashion, right? And what you're noticing, besides where all the phenotypes are like grouped up, is you're noticing where the phenotypes live that are more distinct, right? You're noticing how rare they are out in this, I guess, vast ocean of phenotype space. And then you're also noticing potentially some type of no man's land, right? No phenotypes exist here. What phenotype would you have to produce in order to get one that gets projected down onto this plane in this space, or over here, or over here? Are those feasible? Do they exist? Those go back to the questions that Professor Blue was mentioning. And these are the kind of things that we wish to explore in the future. So I believe we are at 45 minutes. So I'd like to give a couple brief acknowledgements, and then we'll throw it up to public questions. So if you have questions about anything you saw, you can shoot them. Yeah, Patrick.
All right, I was really interested in that cluster you had on the end there.
Yeah.
You take the 960 degree space and represent it to two, I think those are principal components.
Yeah, exactly. Yeah.
I wanted to ask, how did you actually? So like, it's a K-means clustering, right?
Yes.
Okay. How many different variables did the principals component 1 and 2? The different observables in the state did those represent? Is there any overlap? The observables in the platform?
Yeah, good question. So in general, I'm pretty sure you know this as well, Patrick, but the way principal component analysis works for the people who might need a little bit of a refresher are essentially that once you put the data in a matrix, then you can run an SVD on that matrix and then extract the two highest singular values. And then from those singular values, you can, there's associated singular vectors, right, associated with those. And I believe your question, Patrick, essentially is what kind of observables are captured in those singular vectors, right? Essentially. So generally, what I believe is that those singular vectors encode for the time series traces that are most aligned with these 960 dimensional vectors. So for example, you can imagine that if I had, let's say, 2 data points, okay? And I had 2 data points, like 2 time series data points, right? Like time 1 and time 2. Then I have, instead of a 960 dimensional time series vector, I just have a 2-dimensional one, right? And let's say that for both of those data points, right, that I could plot them. I could plot where they ended up, right? And let's say that I was getting fluorescence readouts, right, of like 100 relative fluorescence units, right, on those readouts. Then I would see those pop up in the same location. I would see 100, 100, right, in that location. So if we expand this out and increase the dimensionality, then what we'd see with these types of phenotypes, for example, right, is we would see all of these time series phenotypes kind of like in the same function space, right, for example, right? So what they're encoding for essentially are these types of profiles. They're encoding for the shapes of these guys in general.
It's like time series of different markers.
Exactly.
Okay.
Yeah.
And those aren't like things that are arbitrarily, I guess, quantified. Yeah, this one lit up more higher.
Yeah, exactly. It's essentially just the trace of the profile that took place as vectors, yeah, in this high-dimensional space. Good question, Patrick.
Thank you. Thanks for reminding me about PD.
No worries. Yeah. Yeah. All right.
Great, Dr. Mary. So I'm curious, did you do any kind of analysis on like what kind of preliminary sequences gave you like stronger SFGFP signal? I'm just curious on like kind of that nucleotide sequence grammar.
Yeah. So, correct me if I'm wrong. You're essentially asking what kind of analysis on the genes themselves did we do from the phenotypes? We're working on that currently. Yeah. So far, the main goal was to kind of to just track which gene did what. As for the analysis portion, we've done some preliminary studies of like essentially trying to see what kind of sequences led to what kind of phenotypes. Like, for example, I'll pull up the diagram. So let's take this guy, right? Did a mutation in this location, if it was always a G, did we see the same kind of behavior every single time, right? You could kind of think of that in a way as a type of like gene clustering analysis, right? If are the similar genes cluster in accordance with the similar phenotypes as well. So far, we haven't found good correlations between the same kinds of genes, nucleotide base pairs, right, that you would put in and then also get the same phenotypes because you have so much variability throughout and multiple mutations going on at the same time. That's probably one of the reasons, among others, but also biology in general is very finicky. So yeah, hopefully that answers your question. Right? Yeah. Okay, cool. Yeah.
I have a question as to why you chose folding gate assembly.
Yeah. Yeah, good question. So another method, I believe, is Gibson assembly. I'm sure you know others Melody as well. But essentially, Golden Gate seemed to be efficient at getting the job done. Also, it was like what was on hand and what I was trained in. So oftentimes, it's kind of just like a matter of what's happening at that time, right? Yeah. Also, shout out to Dennis for teaching everything about lab stuff early on. Yeah. Yeah, Pal.
Thanks for the great talk. Could you comment a bit more on the algorithm to detect switches?
Yeah, sure.
You mentioned that you can detect, say, as many switches as there are.
Yeah.
I just wondering, is this like an easy or hard problem? Does it look in the real data? It looks like
Yeah, sure. I'll pull up the slide for the algorithm just for clarity. Okay, cool. So to reiterate your question, just to make sure I'm answering it correctly, Pal. You're saying, the first question I believe you asked was, is this an easy or hard problem in terms of reconstructing the data from the model, or did I mischaracterize it?
Yeah, just knowing like when switches are happening, how switches, like when you get the real data.
Oh, yeah. Oh, good question. Yeah. So Pal brings up a good point that we're kind of like amalgamating several problems at once because switch detection itself is its own can of worms. So this substep right here, part two, I have a backup slide for this one. So yeah, right here, we use binary segmentation, which is one of several switch detection algorithms that exist. But there are several. And once you define a switch detection algorithm in the framework that we defined, then in that algorithm requires you to define a cost function in order for your switch detection algorithm to detect the switches. And then also, switch detection algorithms in general are made just to detect general switches in signals, right? But what I've done is I've adapted switch detection algorithms in a modular way. So that way, if you give me the switch detection algorithm, then I can search over all states of your observables and then detect switches across those states. So you might have seen that. Here's an example right here. right? So this is a 100-dimensional just equation. It's just a vector field equation, right? And you can see switching behavior in the diffusivity, right, of this manifold. And you'll see that we can detect these switches across all of these states. But notice this is for u of t for the state that exists where x is equal to 0.5, but there's literally 100 more states, right, that all got switch detected across, if that makes sense. Yeah. So with that also comes error from each substep as well, because you're getting switch detection error, you're getting optimization error, right? How non-convex is your objective function throughout all of these substeps. These are all things to consider. And these are things that we go into a lot more detail in the paper that I wrote up for this, which you can find out now on arXiv. But yeah, so we also talk about how the error cascades a little bit in the paper. But yeah, good question.
All right, any additional questions? If not, let's thank again, Jamiree.
Thank you.